Our AP Biology or College Biology Unit 2 Cell Structure and Function continues on with 2.9, which is all about mechanisms of transport. Before beginning, let's note the disclaimer below. So we still focus in on the cell barrier, which is the cell membrane, and the way that we are just extending what we previously spoke about, in which we have a cell, and the cell membrane basically controls with a semi-permeable membrane that it is the ability for this phospholipid bilayer, which is embedded with different proteins, to allow the entry and exit of different items such as ions or particles. We talked about this before, but this is just the continuation of what we spoke about before, in which we have membrane proteins. And membrane proteins can be either integral or peripheral. So integral meaning that it takes up the whole space, or peripheral meaning that it sort of lays on one side of the phospholipid bilayer. Now, these proteins can be channels, carriers, or receptors. So a receptor, for example, would be a protein that allows for some sort of attachment. And then we have channel and carrier proteins. And channel proteins are when they allow for some movement, somewhat like a tunnel. And this is usually moving substances from one side to the other, such as areas of high concentration to areas of low concentration. Or we can have carrier proteins, which means that there is some sort of carrying condition of an item through the protein to go from one area to the next. So from the outside or inside of the cell to the outside or inside of the other cell. So making sure that you understand that it's just moving from either inside or outside to inside. So we have spoken about this before, but we're going to talk about it again because there are subtopics associated with it. So previously we talked about active transport, and if you remember, we are moving substances against their gradient. So what that means is with active transport, when you have this protein that's there, you do require some sort of energy, unlike in previous forms of transport, like passive transport. There is some ATP needed, and it's going against the gradient. So basically what you're saying is you use some sort of ATP. So let's say that I put ATP here. It usually shows some sort of energy. This could be light. This could be something related to any sort of energy. They don't just have to say ATP. They could say like it's electrical or something is happening there with some sort of energy, and you're moving against the gradient. So instead of an easy flow, you're going against it. So for example, let's say you're moving substances from lower to higher, and it's not as easy to follow through. It is like we said previously, if you are on a treadmill and you're going in one direction, but the natural flow is in the other direction with the treadmill. You're exerting a lot of energy in general, so that's an easy way to remember active transport. Usually there's a flow in one direction, but you're going against that grain. So we have two separate types of active transport. We have passive versus, or I should say primary active transport, not passive. Primary active transport versus secondary active transport. So these are the two subtypes of active transport. And they both use some sort of energy, let's say ATP, but there is a way that they move that sometimes might be different. So with primary active transport, you usually just have one protein and you're moving particles within that one protein. A sample of this is the sodium potassium pump, where you're using that one protein and you're moving these ions. So one ion would be, for example, Na, and the other one would be K. So you have this one protein and you could be moving Na in one direction, and you could have K also moving, but it's moving in another direction. Basically, it's only utilizing that one protein for assistance. Now, a secondary active transport is using energy from another substance, maybe another protein. So, for example, let's say that you have your original proteins in the bilayer, and then there's some sort of outside assistance to help move the particles along. So, say that you have this cell, this is like one side and this is the other side, but the bilayer extends out, and you see that there's a secondary protein, hence secondary active transport. And maybe they're moving particles in and out of the cell. At the same time, the active transport related proteins are also catching those particles, working simultaneously to assist each other out to ensure the movement of the substance is there. And usually it could be moving one substance on for maybe these proteins and another substance on these proteins, just to sort of compare the balance between the two. That's what it means between secondary active transport. You're utilizing another protein, essentially, another energy-based protein where you are moving particles in and out, and the main proteins are dependent on what's moving in and out by that secondary protein, hence secondary active transport. We also have passive transport, and if you remember, you're going with the concentration gradient. So that would be like the airport conveyor belt. So, for example, you are trying to make sure that you don't miss your flight, and you go along this conveyor belt apparatus and you stand on it and it's moving in one direction and you are also moving in the same direction. It isn't taking a lot of energy or any energy at all, which would be passive transport. And it's just a simple movement. If everything is moving from one side to the other, passive transport just easily moves that in the same direction. Then we had diffusion that we spoke about before, and that's specifically areas of higher concentration to areas of lower concentration. In this picture here, you could see that it's highly concentrated on one side of the cell and not so concentrated on the other side of the cell. And what's going to happen is you're going to have these substances that are going to want to move from one side to the other based on the concentration gradient. So from heavier side to less heavier side. So that's diffusion, and that removes substances from areas of higher to lower. Now, instead of the substances being moving, we can have water that's moving, and that's called osmosis. And we spoke about that before. And with osmosis, you're moving from areas of higher water potential to areas of lower water potential. And usually you see it on the AP biology exam in three forms. But basically, let's say that you have substances that are highly concentrated on one side and not so concentrated on the other. Because we can only move water from one area to the next, we're going to move the water from areas of higher concentration to areas of lower concentration, so the concentration becomes even. So it moves in the direction where it's highly concentrated to remove any sort of disparity. So it becomes equal. So water moves instead of the substances to retain that. We can have facilitated diffusion. So you see this protein here, for example, and maybe that there are substances that are trying to move from one side to the other. There is no use of ATP. It could be the areas of higher to lower. And unlike simple diffusion, there's no protein. Remember, we said facilitated diffusion actually uses a protein. It is a type of passive transport that doesn't require energy, but it can use like either a channel protein or a carrier protein to help assist. Now we talked about the differences between endo and exocytosis previously. So to recap, we can have the idea that there could be some sort of engulfment using some sort of vesicle to have things enter or exit the cell. If it enters the cell, that's called endocytosis. And if it exits the cell, it's called exocytosis. En, enter. Ex, exit. I'm going to first talk about endocytosis. There are three branches. There is phagocytosis, which engulfs really large particles. So you can see here it forms a vesicle, or it's trying to form a vesicle to come inside the cell, and it's engulfing these large particles. If it's a small particle, that's being engulfed in to come inside the cell, that would be called pinocytosis. Think pint. That's how I remember the difference between phagocytosis and pinocytosis. In some cases, on the outside of the almost vesicle, you can have a receptor that exists to help bind, where then it formulates a vesicle to come inside. That's called receptor-mediated endocytosis. They're using some sort of, if you were to take a look at the cell, and then on the outside they're lined with these keys. Once the particle attaches to the key, then it formulates a vesicle type shape to then enter inside the cell. So that's endocytosis. And lastly, we have exocytosis, which is to exit. And if you remember, that's the process of releasing substances from the cell. And it could be small or large, but basically you have these vesicles and they sort of ingratiate themselves into the outside barrier before releasing those particles essentially outside of the cell. Okay, so I understand that this was a brief recap of a previous section we talked about in Unit 2, which is exactly the case. You are building a basic foundation and then further going into the various discrepancies. So with that being said, let's try to tackle a practice question. Take a moment, freeze your computer when you're ready, make sure you read the question and attempt to answer the question. Once completed, we'll review together. Okay, now that you're done reading the question states, which of the following statement correctly describes the process of facilitated diffusion? Is it A, it requires energy in the form of ATP to move molecules against their concentration gradient? B, it involves the movement of water molecules against the selective permeable membrane or through a selective permeable membrane? Is it C, it allows the movement of molecules down their concentration gradient through a membrane protein without the cellular energy? Or is it D, it involves engulfing large particles or liquids into the cell through the formation of vesicles? Some things to note here, I'm going to not go in order to review the answers, but take a moment, pause your video, and once completed, unpause. Okay, now that you're done reviewing the problem, let's see if you got the correct answer. The answer is C. It allows for movement of molecules down their concentration gradient through a membrane protein without the use of cellular energy, which would be the correct answer. Let's go over answer choice A, which talks about ATP, which would lead us to think that there's some sort of active transport involved, which A would be great if we were talking about active transport, which we're not. So I just wanted to point that out there. It involves some movement of water molecules through selective permeable membrane. Because they're talking about water, we're talking about osmosis in choice B, and that wouldn't be correct. And then when we're talking about engulfing, in general, we're talking about endocytosis. But they mention large particles or liquids through the formation of vesicles, so we're talking specifically about phagocytosis. So I just wanted to point out the incorrect answers as well while we're reviewing it. And remember, facilitated diffusion can use a protein, but it falls under the category of passive transport, and therefore it's not going to require any sort of ATP in that case. Now that we're done reviewing those questions, I hope that you like, share, subscribe, because by subscribing, it gives us an opportunity to create more videos throughout the year. Thank you so much.
Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.